Last time we were discussing about nitrification nitrogen cycle. What are the steps involved in that? What types of nitrification is possible? One is atmospheric nitrogen fixation. Another is biological nitrogen fixation. Now, in examination, you have to write a definition of that. How you will write? You simply cannot say that ammonia convert into this form, but you have to write what are the bacteria involved in that, what are the primary product, substrate level, and how it's converting to nitrate form or nitrate form. So first is nitrification. In this process, as you see 2NH3, it means ammonia is converted into nitrate. Nitrate means 2NH3 plus 3O2, that will convert into 2NO2 plus 2H plus 2H2. That you have to remember the reactions. Ammonia is converted into nitrate by the presence of bacteria in the soil. Nitrites are formed by the oxidation of ammonia with the help of nitrosomonas bacteria or their species. Nitrites formed by what? Which process? Oxidation. Oxidation of what? Ammonia. By the help of what? Nitrosomonas bacteria. Later, the produced nitrites are converted into nitrate. This is nitrites. Are converted into nitrates. Nitrates means 2NO3 minus. So NO3 minus by nitrobacter. So two bacterial species are involved. One is Nitrosomonas, another is nitrobot. This conversion is very important as ammonia gas is, you know that it is toxic for plants, but continuously ammonia converted into nitrites, nitrites converted into nitrate. So this is the process of nitrification. Now assimilation. Assimilation, that primary producer means what? Plant take in a nitrogen compound from the soil with the help of their root. So root is taking as nitrogen source in the form of ammonia, nitrite ion, nitrates ion, and ammonia. Therefore, it is ammonium ion. Okay, are used in the formation of plant and animal proteins. So this is the way it enters the food chain when the primary consumer eat the plants. Now ammonification. We have completed. First is nitrification, another assimilation. Now ammonification. When plant or any animal die, then after nitrogen present in that organic matter is buried into a soil, back into soil. So decomposer bacteria or decomposer fungi or decomposer materials or microorganism which will convert into organic matter back into ammonia. This process of decomposition produces ammonia, which is further utilized for the biological process, nitrogen fixation. Okay. Now denitrification. Denitrification is the process in which nitrogen compound make their way back into an atmosphere by converting nitrate into last product of nitrification that convert into a gaseous nitrogen. This process of nitrogen is the final stage and occurs in the absence of oxygen. Remember these words. In nitrification, you require 2NH3 plus 2O2. Here you will not require oxygen. In the absence of oxygen, denitrification is carried out. by the denitrifying bacteria. That species, generally Clostridium pseudomonas, hmm, which will process nitrate to gain oxygen and give out free nitrogen gas. This is the byproduct. Okay, so all about nitrification, assimilation, ammonification, and lastly is denitrification. Now we have completed nitrogen cycle. Now what is the importance of that? We studied only nitrogen cycle, how it is denitrify, how it is nitrification is process, assimilation is process. Then the importance of these nitrogen cycles did that help in synthesizing chlorophyll or chlorophyll from the nitrogenous compound. This is the things you will have to remember. Okay. Another is it helps in converting inert nitrogen gas into a usable form. Third is in the process of ammonifications, bacteria helps in decomposing the animal or a plant matter, which directly helps to clean up the environment. Whatever the material is dyed and buried into a soil are ultimately converted into a soil organic matter. Similarly, nitrates and nitrites. So first nitrites is formed, then after nitrates is formed. So nitrites and nitrates are released into a soil which help or enriching the soil with the necessary nutrient required for the cultivation. It means nitrogen supply. Nitrogen is also integral part of many plant cells and it forms crucial compound that you know very well. All nitrogenous compound alkaloids, amino acid, protein, nucleic acids. So nitrogen is also cycled by the human activities such as combustion of fuels and use of nitrogen as a fertilizer. We are utilizing longer time in making a crop. This process increases the level of nitrogen containing compound in the atmosphere. Continuously, if you are taking or utilizing a fertilizer, it will cause a pollutant in river also, lakes also, and that result into a eutrophications. Now, one by one, whatever we have studied, we have completed up to carbon metabolism, another is nitrogen metabolism. Now the questions of nitrogen metabolism, which is given by Byju's, which is given by online platforms. This is the answer from that. Okay, and our litho too. So first, why do plants need nitrogen? Generally, we think that it is a component of chlorophyll. Consequently, you know very well that photosynthesis reaction require a chlorophyll. Therefore, chlorophyll is the vital process for the photosynthesis. So, the lack of nitrogen that causes a many deficiency disorder in the plant species. So, stunted growth, yellowish of the plant, defoliation of the plants, uh, abnormalities of organs can also be possible with the nitrogen. So, you have to write like this. What are the different steps of nitrogen cycles? So you have to remember Na and D. NAA and D. Nitrogen fixation, assimilations, ammonification, nitrification, and denitrification. Okay. What is ammonification? I already told you that occurs in the decomposition of organic matter, whether it is plant or animals, where ammonifying bacteria convert organic nitrogen into inorganic component, organic into inorganic, that is in ammonification, like ammonia or ammonium ions are removed. What is nitrification? Read.
a process to convert ammonia into nitrate by bacteria. Initially, the ammonia is converted to nitrite by the bacteria Nitrosomonas or Nitrococcus, etc., and then to nitrate by nitrobacteria.
Okay. Now the two things comes. One is nitrites, another is nitrate. As you know, two NH3 ammonia convert into a nitrate, nitrite. First nitrite, then nitrate. So it requires two types of bacteria. So nitrification is the process that convert ammonia into a nitrate by the various two species of bacteria. The ammonia converted into a nitrate by the use of the bacteria that is called as nitrosomonas or nitrococcus. Then nitrate. Nitrite to nitrate by nitrobacter. That you have to write. At least these words comes in your definition, meaning, or what is nitrification. Now, what is denitrification? You cannot write simply back or reversal process. You have to write nitrification. Denitrification is the process of converting nitrate, last product, nitrate back to molecular nitrogen by the use of bacteria, that is Bacillus subtilis, Pseudomonas, Thiobacillus. Hmm. What is the function of denitrifying bacteria? Nitrifying bacteria are a small group of aerobic bacteria. Remember these words, which are mainly involved in the conversion of ammonia into nitrates. Which part of plant is involved in nitrogen fixation? Hmm? Yes, within the nodules, plant root system. Okay. Now, biochemically, how the process is happening? GOGAT, glutamine synthesis. That already been taught to you, even though I am taking here. I am repeating this word. Mitochondria, cytosol, and plastid are involved in this reaction, nitrate assimilation. The assimilatory pathways is here. Is very important physiological process. And major root by here is inorganic nitrogen. Inorganic nitrogen is assimilated into a carbon skeleton in the higher plant, including cyanobacteria, fungi, and algae. So nitrate assimilation is the multi-step process which involves nitrate uptake. Nitrate uptake. That nitrate assimilation is multi-step process. First one. Another, it involves nitrate uptake and is reduction of ammonium by the highly regulated enzyme. Reduction of nitrogen by highly regulated enzymes. There are two enzymes involved in that. One is cytosolic nitrate reductase, another is plastic. Here is cytosolic and plastic. These nitrite reductase, one is nitrate reductase. Nitrate reductase means cytosolic. Nitrite means plastic. So the reduced ammonia. Now reduced ammonia is fixed into a carbon skeleton by utilizing 2-oxoglutarate provided by the TCA cycle, and the reaction is transamination reactions to form glutamate to glutamine. So these two amino acid, which two amino acid? One is glutamic acid, another is glutamate. Okay, glutamate means glutamic acid. Glutamines, okay, that is basic amino acid. Here is acidic amino acid. So these two amino acid act as a major donor. These two amino acid act as a major donor of nitrogen for the biosynthetic pathways of other nitrogen containing compounds and secondary metabolites too. So synthesis of glutamine and glutamate is accomplished or achieved by the two enzymes. One is glutamine synthase, another is glutamate synthase. So GS-GOGAT cycles. Once the nitrate is uptake or taken up inside a cell, it is converted into ammonia or ammonium ion. That is called reduced nitrogen. So reduced form, you know very well in biochemistry, wherever you will find reduced is NADPH. So nitrite or nitrate. Here you hear NADPH NR. NR means nitrate. Nitrate reductase and molybdenum is a cofactor. So cytosolic enzymes catalyze the first and rate limiting committed step in the conversion of nitrate to nitrite. IT to ITE. ATE to ITE. So this enzyme is highly regulated, as I said previously, and all levels of gene expression that may be influenced by many several factors. Okay, many factors. Newly synthesized, you have to remember these words. Newly synthesized nitrite is transported to chloroplast where it is intermediate or converted to ammonia by nitrite reductase, and ammonia is subsequently fixed into organic acid, 2-oxoglutarate, by the combination of this enzyme. These two, one is glutamate synthase, another is glutamine synthase. Glutamine means GS and glutamate means GOGAT. So in leaves, ferredoxine is the major source of electron transport chain or electron for the activity of nitrate reductase and glutamine synthase. So therefore the photosynthesis is the major source of reductant for nitrate, not trait, nitrite. Nitrite and ammonia acetylation into organic acid. Remember this word. Nitrite and ammonia acetylation into organic acid. Although, now take trait आएगा. Although nitrate acetylation can occur only in etiolated leaves containing sufficient carbohydrate. These rates are about two or more than 2 to 5 folds faster in the light. So the condition in green line you have to remember. Now glycolysis. Glycolysis and oxidative pentose phosphate pathways are the process that is involved supplying a reductant and dark cycle or darkness. Hmm? Light may you know very well sufficient carbohydrate is with the etiolated leaves, so nitrate assimilation is occurred. Now the reduced form NADPH generated hexose monophosphate pathways, HMP pathways, has been considered as a significant role in the nitrite assimilation. Where? In the root. So I have taken three things. One is etiolated leaves, another is leaves, third is root. Okay. So where specific form of this ferredoxine NADPH reductase, reduced form, favors the transport of electron from NADPH to ferredoxine. So the amount of photosynthetic electron flow linked to nitrogen assimilation that leak into a leaves largely depend upon nitrogen status. Now another pathways that can also be asked in the examination at the end examination or pathways for assimilation or ammonium assimilation. Now you have to remember here is the structure of glutamine. See, H one side, left side, NH2 another side, COOH. This is the amino acid general structure. Now CH2 two times CONH2 means glutamine. Here is also CH2 two times 1 COH means glutamic. So glutamic acid and glutamate, as I said, glutamine synthetase and glutamate synthase. Okay. Thetase means glutamine. Synthase means glutamate. As I said, pyridoxine 2H plus or NADPH. This is the cycle glutamate to glutamine. Okay. And ammonia uptake, nitrate uptake, nitrogen fixation, and photorespiration. These four cycle, the ATP is generated, ammonia assimilation is there. So ammonia acylation is the central intermediate in the nitrogen metabolism in the plant. Besides, uptake from the soil by the roots. Ammonium is constantly generated in the high rates in the plant, in the tissue, by the process such as nitrate reduction. Another is photorespiration, lignin biosynthesis, and sanations induced nitrogen remobilization and nitrogen fixation in the lignins. So these reactions you have to ammonium is constantly generated in the high rates in the plant tissues, and the process known as nitrate reduction, photorespiration, lignin biosynthesis, sanation induced nitrogen remobilization, and finally nitrogen fixation in the lignins. So irrespective of the source of nitrogen, that is ammonium or organ that assimilated in, there are three parts. One is root nodules, leaves, and roots also. So the key enzymes, as I said previously, two. One is glutamine synthetase, another is glutamine synthase. That is also called as GOGAT. So remember this full form of glutamine oxoglutarate amino transferase. Glutamine oxoglutarate amino transferase. So both the enzyme are present in the roots, in the chloroplast, and you will also found in the nitrogen fixing microorganisms. So assimilation of most of ammonium derived or that taken up ammonium, nitrogen fixations, as I said previously, that nitrate reduction and photorespiration is the mediated by the glutamine synthetase and glutamine synthase pathways. See, this pathway you have to remember. Here is glutamate, here is glutamic acid or glutamate synthase. Okay, here is also glutamate. 1 to 1. Continuously this is formed. So intermediate 2-oxoglutarate, it formed from tetrahydroxy acid cycle, TCA cycle intermediate, 2-oxoglutarate. Okay, and this transamination reaction that will convert into a protein, amino acid, and other nitrogenous compound. So in this pathway, amino acid glutamate act as a receptor, act as a receptor, act as a receptor for ammonium, forming amide glutamate. Now to enzymes, we will study what is their roles, how it is located, where it is synthesized, and what is their roles. Generally, it comes in previously also, but I am repeating. Glutamine synthetase. Synthetase. Remember, glutamine synthetase. That exists in the multiple enzymes, multiple complex. So, located in where? In cytosol and in blastocyst, as I said in the previous picture also. So, cytosolic the glutamine synthetase multiple metabolic functions. Which are the multiple metabolic functions? Already been taught to you. It functions such as assimilation of ammonium ion into a glutamine for transport and distribution throughout the plant. During leaf sanation or etiolated leaves, glutamine synthetase is a key function in assimilation and recycling of ammonium. That can also be asked in the fill in the blanks. What is the function of glutamine synthesis? So, generated from various catabolic process. Remember this one. So, this role of particularly important after anthesis and during grain development, filling in the cereals. That already been taught to you, and I have also told you how the hormones is related with this process. Now, glutamate synthesis. Glutamate synthetase catalyze the transfer of amide group. Amide group means NH2 from glutamate to 2-oxoglutarate, and which is product of tricarboxylic acid cycle, TCA cycle. Now, one by one, we will take what is the functions of that. So, conversion of glutamine to glutamate that take place in the plastids, which have two isoforms of GOGAT. One form accept electrons from the reduced form, or that is paradoxine, photosystem 1, ES1. Other form, that is reduced form NADPH from the respiration. Now, the paradoxine like GOGAT isoform dominate in the leaves, particularly in the chloroplast, phloem companion cell in the leaf vein. Now, NADPH isoform is prevalent in those. So, both forms contain iron sulfur cluster, that is APS cluster, and transfer electron during the reductive stage or reductive synthesis of two glutamate molecules to two oxo to one glutamine molecules and one 2-oxoglutarate. So this is all about glutamate synthesis, and the pathways you can write like this also: GS, GOGAT, and GDH. So one by one arrow is left hand side, another in right hand side. So to produce glutamate is require a maintenance of ammonium assimilation. So high affinity, low affinity. How it is synthesis glutamate, and how if it is high affinity, glutamine. If low affinity, glutamate. Simple conversion. So can be transported from the site of assimilation and utilized by the biosynthesis of protein. So when the ammonia supply is high, both glutamate molecules act as ammonium acceptor. Remember this word. Which condition? Ammonium supply is high. Both the glutamate molecules act as a ammonium acceptor. One molecule of glutamate leaves the cycles. Now the enzyme glutamate dehydrogenase, GDH here. For many years assumed, or it is also many books is also told that, or many evidence is also that is it is involved in the liberation of ammonia during sanation or during defoliation, during etiolated leaves, by catalyzing the oxidation deamination. Oxidation deamination. Deamination means removal of amide group by glutamate and providing carbon skeleton for the respiration and oxidative phosphorylation. Two reactions is involved. One is oxidation, another is oxidative phosphorylation and respiration. So GDH enzyme is the conjunctions. Remember, GDH enzyme is the conjunction with the NADH GOGAT that contribute to controls the leaf glutamate homeostasis. An amino acid that can play a central role in the signaling. And remember this word. It is very important for carbon and nitrogen assimilatory pathways. So, GDH enzyme, glutamate dehydrogenase enzyme, is the conjunction with the NADH GOGAT, and contribute to control of glutamate homeostasis, and amino acid is glutamate, which can act as a central role in signaling or at the interfering of carbon and nitrogen assimilatory pathways. Okay, so we have completed up to GS-GOGAT pathways, glutamine synthesis, glutamate synthesis, okay, GS and GS-GOGAT, their functions, their location. Now urites. Urites are a nitrogenous compound with the special functions that allantoin, allantoinase, that is uric species, and amide, that is glutamine and asparagine, and amides. Okay, so advantages of urites as a nitrogen acceptor or transporter during the lip senescence. Okay, so this is about urites. Now transport of that, the majority of the nitrogen import that take place by outer cotyledons, and one is symplasmically to the cotyledons in the parenchyma cells, another in terminals, vegetative or reproductive sinks such as sink leaves, flowers, fruits to seeds, that nitrogen released from the phloem tissues. That is also via symplectic movement in the parenchyma cells. So this the schematic representation of organic and inorganic nitrogen that already been taught to you how the urease is enzyme and transport of nitrate arrows you can see here, all arrows, nitrate embryo, seed coat, endosperm, source leaf, in the phloem how it is released, seed vacuole, seed loading, phloem unloading and loading, that I will teach you. Okay, so this is the schematic represent of organic and inorganic nitrogen. Arrows from the respective transport reports to the either uptake of nitrogen from apoplast and import into a cell. Okay, so seed vacuole storage and question mark, that unknown transporter still to date in many books. Okay, so how this is involved? These are the certain questions can be asked in the examination. What is chemoautotrophs? Give two example. So you can write bacterial name chemoautotrophs. That is example are nitrobacter, nitrosomonas, and sulfur bacteria. Chemoautotrophs are unique in natures. How? So it harvesting of light energy by photoautotrophs, and chemoautotrophs derive energy from mediated various chemical process. As you know that one is oxidation, another is reductions. So inorganic compound in the either environment, another is nitrosomonas chemolithotrophs, lithotrophs, chemolithotrophs. So as I said, chemoautotrophs, nitrobacter, nitrosomonas, and sulfur bacteria. So nitrosomonas and nitrobacters, now nitrosomonas oxidize ammonia to nitrate. This nitrobacter, nitrate to nitrate, are the best suitable example of these chemolithotrophs. Whether it is this or that, that can also be asked in the examination. So bacteria converting to nitrate, there are the certain definitions, reaction steps, nitrogen cycles, how it involves, what is the end product, what is the substrate, and how the oxygen concentration is involved in the nitrification and denitrification. One by one, we can read this. And temperature, inhibitors. So you have to remember these bases for comparisons of nitrification and denitrification. Some of can be asked in the examination. For example, I am taking nitrification is the process of oxidation reaction. You will find here denitrification is the last step of nitrogen cycle. Okay, so here is the consisting of reduction reactions. Here is oxidation reactions. Okay, ammonia is utilized into nitrites. Here is nitrites to nitrate. Okay, so substrate level is also nitrification process is ammonia. Here is the substrate starting compound is denitrifications are nitrates and nitrites. Okay, so the common nitrogen. This slide is very important. I am not reading this slide. But for examination point of view, for your course content, these slide you have to remember tip of tongues. Any sentence can be asked in the examination. For example, what is the requirement of pH for nitrification? So you have to remember it requires 6.5 to 8. Whereas in case of denitrification, flourishes in the pH range that is 7 to 9. Okay? So you will not find in any lithos. In previous litho also, if you are taking from other persons, that will these basis of comparison you will not find in that litho or any books also. So BNF, this is the general process, biological nitrogen fixation, agriculture system and natural system. Our our governor is depends on this, and he told every person that natural farming is possible. Yes, no. Today it is not possible. Why? Because plants associated this legume rhizobium symbiotic synthesis that is not possible because of continuous utilization of chemical fertilizer that will reduce in the form of associated bacteria. Crop associated bacteria is not present. Therefore, it is not possible to give that particular. So, first of all, what we have to do for natural farming, you require, remember this word, into double inverted comma. You cannot do a natural farming. You require a crop specific bacterial species add into a rhizosphere of that particular plants. You are taking a bacteria from forest and putting into a cereal based product. It is not possible. Natural. So this is the concept these people are not understanding. Natural farming is possible only you have a crop specific bacterial colony. Okay. So agriculture system and natural system. Natural system require a cyanobacteria. You will not find a cyanobacterial population in any soils. Very rare. Cyanobacteria means diatoms. In many books, in Indian people still do not know how many species are in the soil of diatoms. Okay. I'm doing research on that. So free living cyanobacteria, heterotrophic bacteria, autotrophic bacteria, these family is found in all free living. So here is two agriculture system, two things. One is crop, another is pasture and fodder. So plant associated, that is cereal associated bacteria, cereal endophytic bacteria, and cyanobacteria, that is azola and cyanobacteria, that is symbiotic. Legume and rhizobium, that is also symbiotic reactions. So that can also be asked in the biological nitrogen fixations. One has to understand how ultimate biological nitrogen fixation is possible. Conversion of atmospheric dinitrogen, atmospheric gases into ammonia, and finally your soil bacteria that will convert into a nitrites and ultimately nitrates. So nitrosomonas, nitrobacter, both you requires for these biological nitrogen fixations. So this is all about the legumes. Now we have to understand we have completed nitrification, denitrification, assimilation, GS-GOGAT pathways, glutamine synthetase, location of that plastid root nodule zinc. Now in root system we have understand certain bacterial species are the leg hemoglobin. Okay. So the mostly host legumes that senses the chemical produced by rhizobium. These rhizobium core, this rhizobium core, nod factor, nod factor that causes colonized root hairs to curl and forms or In many books, you will get shepherd's crooks, rhizobia that penetrates the root hairs, and typically tubular structure that is called infection thread. Once bacteria reaches the root itself, stimulate the cortical cell division. When bacteria reaches to root itself, cell division occur. Where? In cortical cell. That lead to the formation of nodule. This infection causes nodule. As the nodule begins to form, bacteria become a structure or part of the plant derived membrane and are released inside the plant cell and forming the nodules. So, how nodule is formed, you can write like this. So, bacteria subsequently lose their cell wall and that undergo proposed changes in the cell morphology, form larger, irregular shaped branch cell that is called bacterioles. What is bacterioles? The bacteria infected into a root cells where that undergo profound changes in the cell morphology and that is form a larger one. An irregular shaped debranching or branch cell that is called bacterioles. This all entirely depend on host cell. And in reactor, energy needs, it require energy rate. So bacteria fix nitrogen for the plant. The interaction between the bacteria and nodes legumes is so intricate that particularly rhizobium or bradyrhizobium or will also these genera, selected number of plant genera, only nodulated. Okay. For example, Rhizobium meliloti. Rhizobium meliloti. Only nodulate alfalfa. So certain crop species having a certain rhizobium bacteria and they have a particular name. So that chart you have to remember. As I said previously, why natural farming is not possible? Because the colonization of crop specific nitrogen bacteria is there. You cannot simply taking a soil or some part of soil, some type of urine and fermenting this and putting your pour into a particular plant species. You require these rhizobium meliloti. For example, it is nodulated in the alfalfa, rhizobium leguminosarum or biovar trifoli will only nodulate clover, that is trifolium. So this host specificity is referred to as inoculation group, and that senses cell signaling between the bacteria and legume host. So this is not factor, as I said previously, not factor that causes the colonized root hair, and that is identified as biochemically what is node factor. So biochemically you can say that we identified as lipochition oligosaccharides. Remember this word and repeat this word two times. Lipochition oligosaccharides. Generally, people tells that is protein. No, no, no. This is two parts bacteria and legume host, and the chemical or forced not factor that is lipochition. Here I have given a certain crop species and nodulating bacteria. So you have to remember these lotus, bean, clover, peas. Generally peas and soybean and sesbania that you have to remember. For example, peas, Rhizobium leguminosarum. Then soybean, Bradyrhizobium japonicum, Bradyrhizobium elkani. and rhizobium thread. Okay, certain azorhizobium colinodons. So nitrogen is now enzyme. Nitrogenase. Nitrogenase is the key enzyme for biological BNF, nitrogen fixation. So now you understand what I am telling. Why natural farming is not possible? Crop specific bacteria is required. You cannot simply taking a night soil or podras, or you can say cow dung seed, and putting into making a solution of gomutra and what they are telling jivamrut and panchamrut. It is not possible. Why not possible? You have to understand the nitrogen fixations in the plant, as well as you have to understand the carbon and nitrogen assimilation in the plant. So nitrogenase is the key enzyme for the biological BNF. Which is the key enzyme for biological nitrogen fixation? Nitrogenase. So nitrogenase convert the nitrogen to ammonia, and it is present in the some prokaryotes. Reduction of this dinitrogen N2 to ammonia by living organism called biological nitrogen fixation. So nitrogenase catalyze the conversion of nitrogen to ammonia, and ammonia is the first stable product in the nitrogen fixation. So, which is the first stable product in the nitrogen fixation? Biological nitrogen, ammonia. Nitrogen act as a catalyst and reduce the activation energies. So, reaction you can see here. Okay. Remember N2, you have to write like this, huh? Not like this. Control plus. Okay. So, NH3, 2NH3, you have to write like this. 2NH3 plus H2 plus 16 ADP plus 16 inorganic phosphate. Okay. Sola, ammonia plus H2. So nitrogen plus 8 electron plus 8 hydrogen plus 16 ATP. 16. That will is the key enzyme for biological nitrogen fixations. Some of the important characteristic of nitrogenase enzymes, that is nitrogen is present in the prokaryotes. You have to remember this word. It carry out nitrogen fixation in the living organism. Nitrogen is convert atmospheric nitrogen to ammonia. It is a MOFE protein, heterotetramers consisting of 2 alpha and 2 beta subunit, and oxidation state of MO, and APS cluster between alpha and beta subunit. These are It is highly sensitive to molecular oxygen. It is highly sensitive to molecular oxygen. Nitrogen enzyme require anaerobic condition for its activity. And leg hemoglobin present in the nodules. Leg hemoglobin present in the nodule act as oxygen scavenger. Scavenger oxygen को remove करेगा and help in maintaining an aerobic condition, that is required for what? Nitrogenase complex, nitrogenase enzyme for the biological nitrogen fixation. And it requires, as I said previously, 8 electron plus 8 H plus 16 ATP, that will convert into ATP required for the conversion of nitrogen to ammonia and from the respiration of the or cells. Now nitrogenous enzymes types, that is vanadium types, iron only, or molybdenum nitrogenase. That is a various type of nitrogen fixing bacteria. Three types of nitrogenase found in the nitrogen fixations. So key enzyme for sustainable. Remember, these nitrogenase is very important for life on Earth. Which enzyme? That can also be asked in the biological nitrogen fixation. Yes, but you require these for what? It is the key enzyme for sustaining life on the earth. Okay, so nitrogenase complex you can see in the picture also. Fe protein and MOF proteins, dinitrogenase reductase and dinitrogenase. So the diagrammatically structure of nitrogenase enzyme complex. and I have already been in some species of azotobacters. Okay, so certain culture is also available in the market for leguminous crops. Okay, so general structure and mechanism of this heterodimer consists of two alpha and two subunit and MOFE protein component of these nitrogenase, denitrogenase reductase, and this is the all about biological nitrogen fixation. You can read. Next gene we will takes afterwards. You tell me. Okay. Thank you all. Is there any questions? Hmm? There is. Pause or here. Here.